And so with that, we'll go ahead and get started. So I'm going to start with a very basic principle, and that's that we're all human. So since we're all human, we have virtually the same DNA. So 99.9% of our DNA is identical between any two individuals. But that 0.1%, that small bit of variation, is important. And that small bit of variation creates changes in height, in personality, predisposition to disease, and also what we're interested in for imaging genetics, brain structure, and function. So this is the outline of what I'm going to talk about. So first I'm going to go over what types of genetic variation exist in the human genome. What is that 0.1% that makes us all different? Then I'm going to go over some big questions in imaging genetics that I feel are important, but this is by no means comprehensive, and you may feel other ones are important. We'll go over heritability, which is motivation behind doing genetic search. Then tell a little bit of history about imaging genetics. And then what is our progress so far in answering some of these big questions in imaging genetics? Okay, so let's start with some very basic human genetics. So with some terminology first. So first we have 23 pairs of chromosomes. A chromosome is just a long strand of DNA. And you have pairs. You have one chromosome from your mom and one chromosome from your dad for each of those 23 pairs. If we zoom into that chromosome and then we sort of put it over on its side, we can zoom in to where this red box is, and we can see that within a chromosome there are many genes. So genes are annotated here in this portion of the graph. And then within a gene there are different features. There are exons here and introns, and there's also single base pair changes called SNPs, or single nucleotide polymorphisms, which I'll talk about more. And a SNP is a site in the genome that's variable across people. So here you can see that you have one person that has the C allele and another person has the T allele. So to go a little bit more in depth into this, to make sure that we are all on the same page with the terminology. So you inherit chromosomes from your dad and from your mom. So you have pairs of chromosomes, and most of your DNA is similar or is identical between any two people. But at certain places in the genome, like here, there is variability across people. And so those places where there's single nucleotide changes across people, we call those single nucleotide polymorphisms, or SNPs. So here's an example of a SNP where this person has an A allele and a G allele, which gives them a heterozygous genotype. And here's an example of a homozygous genotype where they have an A allele and an A allele. Now, say I conduct an association study and I try to see do people with the G allele more often have a neuropsychiatric disease? And so if that is the case, then we call G allele the risk allele, whereas A allele is the protective allele. G allele creates risk for the disease. A allele, it protects from the disease. Okay, so another piece of terminology that's commonly used in the field is either major or minor allele. So here's what this means. So say I measure a single nucleotide polymorphism. I measure the alleles across a large population. And then I see that the C allele is present in 70% of chromosomes, and the T allele is present in 30% of chromosomes. That makes the C allele the major allele. It's more than 50% of the population, whereas the T allele is the minor allele. It's less than 50%. Okay. So that's kind of the basics of SNPs, of the most common variation that exists in the genome. So how many variants actually exist in the genome between any two people? So this has been measured by a consortium called the 1,000 Genomes Consortium, and they've done whole genome sequencing on over 1,000 individuals, I think almost 2,000 individuals. And they find that the number of variants per person is about 4 million, depending on what your ancestry is. Africans actually have more genetic variation compared to the reference genome. So each individual has 4 to 5 million variants that are different from the reference genome. And that 4 to 5 million divided by around the 3 billion gives you that 0.1% of variation. That's how each human is different from every other human. So almost all of that variation is SNPs, are these single nucleotide changes, these single nucleotide polymorphisms. So 99.9% of those variants are either SNPs or short insertions and deletions, which are called indels. So most of the variation in the genome is small structural or small sequence variation. But that's not to say that that's the only type of variation in the genome. In fact, that there's many types of variation in the human genome. The genome can be messed up in many different ways. Okay, so it can be messed up at the sequence variation, the individual base pair level. so single nucleotide changes, and those are, an example are single nucleotide polymorphisms like we talked about before, or indels, insertions, or deletions. There's also structural variation, which can be sort of larger. It's on the multi-base level. And that can be many different types of variation. You can have repeats of sequence, you can have inversions, you can have copy number variations where you have deletions or duplications. So there's a lot of different types of variation. There's a lot of ways that the chromosomes can change. Some of them are really interesting. You can have ring chromosomes where there's the chromosomes actually attached to each other. So these are much rarer though, and the most common type of variation is the sequence variation. You have different methods for detecting these types of variations. So molecular genetic detection, so mainly genome sequencing, is used for these single nucleotide changes. Whereas if they're big changes like different numbers of chromosomes, for example, then you can detect them with a microscope, cytogenetic detection. Okay, so you can have this variation in the genome, and it can have different functionality. So if that variation exists in the exome, or in the coding regions of the genome, it can damage the proteins or alter the structure of proteins resulting. So if you have a mutation in the protein coding sequence, it can create a change in the amino acid structure of the protein and result in what's called a missense mutation. There can also be insertions or deletions. In this case, you have an insertion of an A. The insertion of an A often leads, or insertions often, or deletions often lead to frameshift mutations, which are early nonsense mutations. So they create new stop codons and the protein isn't fully made. There's also splice site mutations, and then most of the genetic variation that we talk about and that we know about is intronic or intergenic variation, which is 97% of the, which is because 97% of the genome does not code for proteins. So Bernard's specifically going to talk about this intronic or intergenic mutations because these often are thought to have a gene regulatory function. So they regulate the expression of different genes. Okay, so when you're born, you inherit chromosomes from your parents, but those chromosomes are not just copied, they're actually sort of mixed between your parents and then passed down. So this mixing of chromosomes from your parents is created during meiosis through crossing over or homologous recombination, and it keeps happening. So you keep getting these crossing over points. And so as you're creating this mixing, it has the effect that genetic variants are inherited in chunks, such that there's a correlation between alleles at nearby SNPs. So this is a very important feature of the genome because you get chunks of variation that are inherited together. And that means that when you measure a particular genetic variant, you're not just measuring that variant, you're also measuring the variants that are correlated with that variant. Okay, and one last piece of information about genetic variants is that mutations in the population happen and sort of can get different frequencies in the population. So this is the allele frequency, and this is, you can think of it as time in generations. So a mutation happens sometime in the population. That mutation can undergo random genetic drifts because certain people have more babies, certain people have less babies, and then that mutation can be advantageous in the population and then can shoot up in allele frequency, for example. It can also shoot down in allele frequency. If it shoots up, it's called positive selection, down called purifying selection. So we generally don't know the evolutionary histories of each genetic variant in the population, but this is an important thing to keep in mind. Genetic variants that we study, like common variants, have been sort of up and down in the population for a long time. De novo variants, which is another type of variants, have only existed for a very short time. Okay, so all of the genetic variation that I talked to you previously about, we sort of have made the assumption that the same genetic variation exists in all cells in the body. This is roughly true, but it is not completely true. And so when it is not true, it's called somatic variation. So in this case, you have an egg and a sperm, and it forms a zygote, but during development, there can be a mutation that happens. And then after that mutation, all the cells will acquire, all the cells derived from this cell will acquire that mutation as well. So this is a type of what's called somatic variation. It's not found in every cell in the body, it's only found in certain cells in the body. So this has only been very recently profiled in human neuronal cells or human brain cells. And what we find is that there's basically around 1,400 somatic mutations from single-cell genome sequencing. So the genome is not actually the same in every cell in the body. It's slightly different. Most of that difference is found in these intergenic regions because that's most of the genome, although some of it is found in these coding regions and can be, for example, missense mutations, but a very low percentage, like 0.8%. So it's important to know, though, that brain-affecting somatic mutations will not be passed on before the development of the gonads. Okay, so this is a quick summary of all of the genetic variation that exists in the human genome. So everyone has different genetic variants, but most of those genetic variants have an infinitesimally small effect on the structure of the brain or on any trait that you're looking at. So really the whole goal of imaging genetics is to identify which of those genetic variations affect brain structure and function. So that's like the overarching goal of what we want to do. If there's much genetic variation that exists, which ones can we focus on? Okay, so that leads us to why do we want to do this in general? Why do we want to find genetic variation that impacts the structure or the function of the human brain? So here are some questions that I think are important in this field that I think that we have the ability to make headway into. So first, how does genetic variation lead to disorders of the brain? This gets to the end-of-phenotype concept, the idea that genetic variation impacts brain structure and then leads to neuropsychiatric diseases. So we can basically assess that using imaging genetics. A second thing we want to do is to identify what cell types or developmental processes impact brain structure. Do genetic changes that affect brain structure also affect brain function? And if they only affect brain structure in the absence of affecting brain function, do we care? This is another question. So what genetic changes also have led to humans having big brains and unique cognitive capabilities? So this is an interesting question too about human evolution. Okay, so before we get into the progress of those, I want to just give you some brief history about imaging genetics and motivate doing the genetics more. So the first question we want to get to, does genetic variation impact the structure or function of the human brain at all? And to ask this question, we usually go to heritability. So heritability asks what percentage of trait or phenotype variance is due to genetic variance. If heritability is 0, there's no reason to do a genetic study. If heritability is high or just significant, just any number above 0 basically, then there is some genetic variation influencing a phenotype. So the way that this is usually measured is through twin studies. And the idea behind this is that identical twins, which are monozygotic twins, they share 100% of their alleles and a common environment. And fraternal twins share, on average, 50% of their alleles and a common environment. So the goal here is to say, is brain structure or brain function more similar between identical twins than it is between fraternal twins? If so, then we have some evidence for a genetic contribution, and that's called heritability. So with that, is brain structure heritable? And so here's, I think, a really nice summary of a lot of different brain structures, showing that basically if the brain structure is large, so volume is on this axis and heritability here, so if the brain structures are large, then you generally get higher heritability. Smaller brain structures generally have smaller heritability. And that's most likely because there's noise in measuring smaller brain structures. The higher the noise is, the less genetic variation that you see. So heritability calculations are highly susceptible to noise because you decrease the correlation between twins. So most well-measured brain structures are heritable. And probably if we were able to measure these well, then they'd all be heritable. Although I don't have evidence to show that. Okay, so that shows that brain structure is generally heritable. What about brain function? So this has actually been assessed recently in the UK Biobank study, and here they calculated heritability in a slightly different way, but they assessed whether brain structure and brain function have what the level of heritability or the level of genetic contribution to a trait is for structure and function. And generally you see pretty significant, about 50% heritability for most of brain structure, but brain function surprisingly lower. And this is, again, most likely because there's higher errors, higher measurement errors in brain function measures than there are for brain structure measures. Okay, so now a brief foray into the history of imaging genetics. And so we'll start with what I call the dark ages of imaging genetics. So there were some assumptions made in the field before we started conducting these really large sample size, big genetic associations. And those assumptions were that measures of brain structure and function are closer to the action of the gene than behaviorally defined neuropsychiatric diseases. So closer to the action of the gene means that they have a higher effect size. So genetic variants have a higher effect size on brain structure or function than they would on the disease itself. Okay, so the second assumption was that measures of brain structure and function are genetically correlated to neuropsychiatric diseases. So what that means is that the genetics that influence brain structure are the same as the genetics that influence risk for psychiatric diseases. So these are not bad assumptions, and they may or may not be true, and we can evaluate them, and I'll show you we do evaluate them now. But what the implications of these assumptions was that we need smaller sample sizes to detect genetic influences on brain than on behavior. And the other implication was that genetic influences on brain will be informative about genetic influences on behavior. So we can assess whether these are true, but let's focus on the first one first. So we need smaller sample sizes because we assume there are higher effects. Okay, so one of the first studies to do imaging genetics looked at amygdala response to based on different groups of a serotonin receptor, different genotype groups of a serotonin receptor. And they found that the effect size of a common variant on brain function was about 40%. So 40% of the variance explained. So the sample size they used was small, about 14 subjects in each group. And the gene and the variant chosen were based on prior biological knowledge. The serotonin transporter allele has something to do with the function of the amygdala. Okay, so studies like this have unfortunately not been very well replicated. The basics of that are, the reason behind that were that the effect sizes were hugely overestimated. So current effect sizes in well-replicated studies are well less than 1% of the variance explained. So what that implies is that the literature prior to around 2012 was full of a lot of false positives. So the literature of imaging genetics contains many false positives. And here's a bunch of references that I think are very useful for the field. So, and that was, again, because we assume that there's high effect sizes for imaging genetics findings, but there are in fact, pretty low effect sizes for imaging genetics findings. So the second, what that led to is an acknowledgement that we're very bad at guessing what the particular genes or variants are that influence any biological process, whether that's neuropsychiatric diseases or brain structure. We can't pick candidate genes very well. And so what that means is that we need to then scan across the entire genome without any biases to try to find any genetic variants influencing whatever our trait is. So what that led to is the development of uniform significance thresholds where everybody has to use the same significance threshold. So for common variant associations, those genome-wide significance thresholds are p is less than 5 times 10 to the negative 8. That's because the LD structure of the genome gives about 1 million independent pieces of the genome that we're testing, no matter what kind of common variant imputation that we do. And we divide 0.05 divided by 1 million, and we get 5 times 10 to the negative 8. So that's roughly how this was calculated. This is the threshold that people have been using ever since. And because of this, things have started to replicate pretty well. So also replication and meta-analysis were required. So nobody had enough sample size to get significance, especially with effects of this low, because you need basically around 10,000 subjects as sort of a gold standard in GWAS studies. So meta-analysis was used and replication was used, and Paul's going to talk a lot about that. And basically after this, now we have associations that replicate. So I would encourage you, when you guys are doing your imaging genetics research, please use this threshold, make considerations about your sample size, knowing that effect sizes are, in general, well less than 1% of variance explained. Okay, so now I'm going to talk about some of the progress in imaging genetics for replicable findings, for well-replicable findings. So there's different methods. There's different methods for doing genetic association or doing genetic linkage to identify genes influencing the structure of the human brain. So those methods are based on both the allele frequency and the penetrance. So if we have very rare alleles that are only found in small families that have really large effects, then those are Mendelian diseases, and we can find them through linkage, which is sort of an older school method that's not really as much used anymore. alleles are very common in the population. Those common alleles probably don't have a huge effect on the structure of the brain or on any particular phenotype, because if they did, they would be selected out of the population pretty quickly. So, so for those, they have a low effect, and we assay those using genome-wide association studies. And then we have sort of the middle category where we have low-frequency variants with intermediate penetrance, and those are done generally with genome or exome sequencing or also copy number variant calling that Carrie Bearden's going to talk about. So it's important to know what doesn't really exist very often either. So one of those is common variation with high effects. So if a common variant has a very high effect, we wouldn't expect that to really exist in the population for a long time. It would be selected out of the population generally. There are some exceptions to this, especially with late onset diseases like Alzheimer's disease. So for example, ApoE is a common variant with an unusually high effect. So there's very few examples of this, though, especially in psychiatric diseases. And then if a variant has a very rare allele frequency and has a very low effect, you can imagine statistically it's very difficult to detect that. Very difficult to detect something that has a very small effect. Okay, so let's talk about some of the rare variants associated with human brain structure. So the biggest ones, the ones we really know really well, are those associated with microcephaly. And microcephaly is basically just a overall shrinking of the entire brain, and mainly the cortex. So rare recessive variants have been identified through linkage. They've been predominantly affecting centrosomal proteins, and they can lead to primary microcephaly. Okay, and here I wanted to give you one example of a microcephaly-associated genetic variation. So it was detected in a family in Saudi Arabia that the patients exhibited microcephaly, polymicrogyria, which is a bunch of little gyri, and agenesis of the corpus callosum, the crossing fibers. And so through linkage analysis, they identified that there was a translocation near a gene that's really developmentally important called TBR2. And so what that means is a translocation is that you chunked off a bit of this chromosome and put it on another chromosome, and took that chromosome and put it here. And so there's basically a difference here. So you actually removed regulatory elements that are governing the expression of TBR2 from this gene. And when you do that, at least in blood, you get absolutely no expression of this EOMS gene, here. So what this identifies is not just a genetic variant, but also a gene that impacts the structure of the human brain. And this is a very highly interpretable gene because we know it's involved in the development of the human cortex. So that's just one example of this category that we talked about. And now I'm going to move to here, this genome-wide association category. So we measure SNPs differently with genome-wide association. And the basis of doing a genome-wide association is that there are many genetic variants that exist in the human genome, some of which are common. And so most of the genome is identical between any two people because we're all human, but there are some places where there is variation across people here. And if we compile all where those variants are, there are certain SNPs that are correlated highly with other SNPs nearby. And those are called tag SNPs. And so genome-wide association studies only genotype those tag SNPs, but are able to impute and to get the other SNPs because of the linkage disequilibrium structure of the human genome. So there's correlation with these other SNPs, so you only need to genotype a subset of them to get the entire genome. So rare variants are less correlated with common variants, so this technique only works for common variants, and that's why this is a common variant association technique. So how does it actually work? So we can purchase SNP chips, which you can purchase from basically two different companies. And in these companies, they take genomic DNA, they amplify it, they fragment DNA into little pieces, and then they wash it over these chips here. The chips have little sequences of DNA that are bound by beads, and then you can add a polymerase with a fluorescently labeled new nucleotide. And so that single base addition flashes, and you take a picture of it, and that's how you know what the genotype is. Now, the nice thing about this is that arrays are about $100 per sample right now. You can get them even cheaper. There's basically like $50 per sample. It's really cheap. It's way cheaper than an MRI. You know, an MRI is like $500 per sample. So you can really get these quite easily. Now, whole genome sequencing, where you're actually sequencing every base pair, is now around $2,000 per sample. Depends where you go. You can get this to $1,000 per sample. We're definitely at the $1,000 genome, at least when I get emails for specials, at least during specials. So this one, the arrays are actually much cheaper now, but the cost of genome sequencing is actually going much down, or down quickly. So Paul's going to mainly talk about this, but I just wanted to highlight several consortia that are doing common variant associations to brain structure. Again, consortia were needed to get enough sample size to identify significant effects for very low effect size variance. So with around, so those consortia are Enigma, Charge, UK Biobank, and they've all have led to replicable results. And so here's just one example of that. With around 33,000 subjects, we detect several genome-wide significant variants influencing intracranial volume, the overall size of the head. And looking at just one of those genome-wide significant variants, it's near a gene called IGF-1, which is a growth factor, that when deleted in mice really shrinks the size of the brain. So this is just one example that's interpretable. I made an assumption here that this intergenic variant is actually affecting this gene, which I have not proved. But in general, we have identified a genetic variant near a gene that makes sense in terms of overall brain structure size. Okay, so now I've talked about Mendelian diseases, genome-wide associations. Now talking about this sort of newer field, where we do genome exome sequencing or CNV calling from genotyping chips. And so this is, again, to detect low-frequency variants with intermediate penetrance. So the main way of doing this is using what's called next-generation sequencing or short-read sequencing. In this, you get your DNA from your sample again. You fragment that DNA into little pieces, you add adapters to those pieces, and you wash it over a flow cell, which contains a bunch of these adapters that hybridize. Then you can amplify those little pieces of DNA, and then you can add a nucleotide with a fluorescent label to each of those sequences. It will fluoresce, you take a picture, you cleave it, and then you go to the next one. So you can keep adding them on, and then by doing this, you're getting the sequence of all of these different pieces of short reads. You can then map those short reads back to the genome. So this is definitely more expensive than genotyping chips. Again, costs are going down now, but it allows you to genotype every base pair. So instead of genotyping just a subset of base pairs in the genome, here you're going to genotype every base pair. Okay, so there's also a variant of genome sequencing called exome sequencing, which allows you to focus in only on those coding regions of the genome that are currently the most interpretable. So we understand the interpretation of those better. So in this, you get your genomic DNA, you again fragment it, but then you have these probes that you've designed that pull down pieces of the DNA that are in the exome. So you've already designed these to pull down pieces that are in the exome. You can sequence them and then map them back to the genome. So this is a way to enrich, and you're sequencing less here. So it's definitely cheaper. I think it's around $500 or so. Maybe Sven knows more, but $500 or so for exome sequencing, and you're getting only 3% of the genome now with exome sequencing. Okay, so how do you use this sequence-based information to identify genetic variants? Well, you can use it to identify multiple different types of genetic variation that we talked about before. So here is the reference genome. And then you can see that all of the reads here have a different allele. So you would have a homozygous point mutation here with the C allele. You can also identify insertions or deletions by breaks in the mapping. You can identify homozygous deletions just by no reads mapping to a certain location. Hemizygous deletions or gains by seeing the number of reads mapping. And also translocation breakpoints by identifying if a read maps to chromosome 1 and then also maps to chromosome 5. In addition, a new thing that people are doing now is to look at pathogens or non-human sequences to try to see if there's, the genetic variants that are in your DNA, are they belonging to other species, like bacteria or things like that. Okay, and then how do you conduct an association test using this type of data? Generally, it's done using what's called a burden test, where you could get cases or controls, or people with big brains, people with small brains, for example, and then you aggregate all the mutations that are of a certain type. Maybe they disrupt genes or missense mutations, for example, across a feature like genes, and then you count, and then you do a statistical test to see if there's a significant difference between your people with big brains and your people with small brains. Okay, so this is pretty new technology and still kind of expensive. So there are actually very few examples that I know of that do this whole genome or exome sequencing in imaging genetics. So this is surely a burgeoning field, and there are many more examples soon to come, and I'm sure some of you guys are working on this. Okay, so now I've went through what types of variation exist in the human genome, motivated that a lot of the different brain structures and also to a lesser degree brain function are heritable, so there must be genetic variants impacting the structure of the brain, and then how those variants have been found and which variants have been found. And this will be gone into greater detail by Paul and Carrie. And then, so now getting to what is the progress in answering some of these big questions that I went over before. So first question, how does genetic variation lead to disorders of the brain? So this is often thought of as what's called the endophenotype concept. And so the endophenotype can be prioritized along two axes. So axis one is genetic correlation with the disease. So do the same genetic variants that influence the structure of the brain also influence risk for disease? So if this is high, and those genetic variants have a high effect size, therefore I need a smaller sample size to detect them, then that's what I would call an optimal intermediate or endophenotype. So that would be something that I can use to detect and find new genetic variations associated with diseases. Okay, so how do neuroimaging traits fare in this? So go over that 1 second. So, but before that, so genetic correlation implies two models basically that you can use. So one is called the mediational model, and this says that your genes influence your endophenotype or brain structure or function on the way to influencing disease. So this is a causal relationship. The genes that influence your endophenotype also influence risk for disease. So this is called the mediational model, and it will help you do mechanistic study and also higher powered genetic search. Okay? So it will help you understand how genes create risk for a psychiatric disorder. Another model is called the pleiotropy or liability index model. So in this model, the genes independently influence your endophenotype and your risk for disease. Okay? And so in this model, if I vary the endophenotype, if I change brain structure, it actually doesn't tell me anything about disease. So they are not causally or mechanistically related. So in this model, I don't get this mechanistic thing, but I do get, if genetic variants have a higher effect size than they, on brain structure than they do on disease risk, then I get this part, that I get a higher powered genetic risk. So it's important to know that we have not been able to separate these two models yet, but we really want this model. And this has been what is sold in the community, so that's important. Okay, so so these are what makes an endophenotype. So how well do we do? So first we're going to talk about the effect size axis. So here's from our recent paper looking at effect sizes, which is graphed on the y-axis, for different SNPs associated with either brain structure, height, schizophrenia, or educational attainment. And what we find is, first, that common genetic variants have a very similar effect on brain structure, effect sizes on brain structure as compared to neuropsychiatric disease risk as compared to height. Okay, so we are not by any means in sample sizes now approaching like 30,000, 40,000 subjects. We are not getting effect sizes of 40%. Okay, we're getting effect sizes much less than 1%. Some of those are actually surprisingly large though, and some of them are slightly higher than what you would get for height or schizophrenia. But still, when you're designing your studies, your imaging genetic studies, you want to take this into account. Your effect sizes are going to be less than 1%, or you should assume that they will be less than 1%, even if you're studying some kind of new phenotype that we have not yet studied. Okay, so now going to the second piece of this, the genetic correlation with disease. So a previous assumption was basically that most neuroimaging traits will be genetically correlated with disease. So the same genetic variants that influence the structure of the brain also influence your risk for psychiatric diseases. So we can actually evaluate that now using what's called genetic correlation. And this is a genetic correlation between intracranial volume, the overall size of the head, and different neuropsychiatric diseases. And unfortunately, the genetic correlations are all right around 0 and not significant. So that's for autism, bipolar disorder, major depression, schizophrenia, extroversion, neuroticism. So this is kind of unfortunate. The genetic variants that create changes in the overall size of the head are actually not the same as those creating risk for these psychiatric diseases. Now that may be due to low power, But it does imply that these effects are not going to be huge. They're not going to be really large genetic correlations. Now, that is not always the case, though, and we have at least one counterexample where genetic variants associated with decreased hippocampal volume are also associated with increased risk for Alzheimer's disease. So this is a nice example. It's kind of what we expected. So genetic variants in the general population that decrease the volume of your hippocampus are increasing your risk for developing Alzheimer's disease. So that's a nice positive example and may indicate that hippocampal volume is a good endophenotype for Alzheimer's disease. But here, again, is an example with schizophrenia and looking at multiple different subcortical structure volumes. And the genetic correlations with these are all also pretty low and none of them significant, despite the fact that there are differences in volumes between cases and controls. Okay, so let's review. So how well are brain structure, at least the brain structures that we measured here, how good do they model this endophenotype diagram? Well, they're okay, not as good as we expected for sure. So maybe the best one so far is hippocampal volume with Alzheimer's disease. That's not to say that these, so other brain structures that will be measured in the future or other brain functions may exhibit higher effect sizes and may exhibit higher correlation with disease. We don't know yet, so we'll see. Okay, so now let's talk about what cell types and developmental processes impact brain structure. So I'm going to focus on this, on talking about genetic variants associated with intracranial volume or the overall size of the head. So with intracranial volume, it's mainly driven by cortical volume because the cortex is a really large part of the brain. And so here you can see the development of the entire human brain from 23 days post-conception all the way up to birth, here, newborn, all the way up to 90 years old. And here are some of the developmental processes that are going on during the development of the human brain. So during fetal development, almost all of the neurons of your entire brain are born. So your neurons are born before you are. Astrocytes are born after that, and then oligodendrocytes continuing into childhood. Synapses, myelination, and synaptic pruning are all happening later on. So our question is, which process, so we have genetics that impact intracranial volume. Are those genetics impacting neurogenesis processes, or are they impacting synaptogenesis processes? How are they changing the structure of the brain? So to assess this, we can first look back in the literature about some hypotheses about how does brain structure change. So one of those hypotheses is called the radial unit hypothesis. And in this hypothesis, there are multiple different proliferative neural progenitor cells that create the neurons of the cortex. So these are only present during fetal development, during this time here, prior to birth. So if I increase the number of proliferative neural progenitor cells, I get an increased cortical surface area in this hypothesis. And if some of those neural progenitor cells die, so maybe they have premitotic cell death from apoptotic factors, then I get a decreased cortical surface area here. So this radial unit hypothesis says that the more proliferative neural progenitors you have, the bigger your cortical surface area. So to assess if the radial unit hypothesis applies to genetic variation involved in intracranial volume, we measured chromatin accessibility in the developing cortex in these neural progenitor cell area called the germinal zone and in the cortical plate. So the cortical plate contains the neurons that are derived from these neural progenitor cells. So we measured chromatin accessibility, which is a way of assessing what is the function of the non-coding genome. And we can assay that across the entire genome in these two tissues. And what we found is that there's enrichment and heritability explained in the regions of the genome that are associated with intracranial volume for these GZ peaks, the ones that are involved in neural progenitor proliferation. So what this implies is that genetic variation impacts neural progenitor regulatory elements during fetal development to impact adult cortical surface area. So actually the genetic changes impact during fetal development to result in changes in the adult cortex, in the adult brain. Okay, and then the last question that I'm going to go over is what genetic changes have led to humans having big brains and unique cognitive abilities? So just quickly, humans have big brains. So here's a human brain and here is a mouse brain. They are very different. So a human brain has around 170 billion cells, and a mouse brain has around 100 million cells. The biggest difference between human and mouse brains are that human brains have a 1,000-fold bigger cortical surface area, whereas they only have a 2-fold bigger thickness. So the surface area is greatly expanded in humans as compared to rodents. Surface area is also expanded compared to our closest living relative, the chimpanzee, versus the human. Through fossil evidence, we found that from 2 million years ago, so humans separated from chimps around 6 million years ago, it's estimated. And then from around 2 million years ago to 200,000 years ago, humans have increased the size of their brain. So the endocrinial volume has increased in humans. And then over the last 200,000 years, there's been a great increase in the size of the brain. And actually around the last 50,000 years, there's been a slight decrease in the size of the human brain. So the human brain size is definitely changing even across human evolution, and it's changing. It's definitely changed across different species that exist. So now, how do we detect or create hypotheses about which genes are involved in human evolution? Well, we have these unique data sets now, where we have data sets of how genetic variation is influencing the structure of the human brain. We also have data sets that tell us where in the genome there's been evidence of selection or changes across different species, or even changes within humans. And so we can combine those two to create hypotheses about the genetic basis of evolved brain phenotypes. So this is really new, and if you want, I have a poster on this that you guys can come see. But this is, I think, a new direction and a really interesting one for the imaging genetics field. Okay, so some overall conclusions. Each human genome has a lot of variation. All of us have a lot of different genetic variants. Only some of that genetic variance creates changes in the structure and function of the human brain. Modern genomic methods and large sample sizes have allowed for this discovery of genetic variants influencing human brain structure, and importantly, the replication of those variants too. And the recent explosion of well-replicated genetic variants associated with brain structure will allow new insight into human evolution, neuropsychiatric disease risk, and brain function if we can interpret them. And our other guys will tell us how to interpret them. Okay, so that's my talk, but I wanted to give you guys some questions to make sure you understood some of the stuff that I was saying. So the first question, what is the difference between an allele and a SNP? Anyone, just shout it out. A SNP is single nucleotide polymorphism. Good, and it is a location in the genome. So an allele is the variance of the state of that SNP. Good, thank you. Why do we use a uniform significance threshold in a genome-wide association study? Confirming correct. Multiple comparisons across 1 million regions of the genome. Things replicate after you use this statistical significance threshold in general. So most things have replicated with this threshold. What are 2 important criteria for a good endophenotype? Large effect size, good. And then the other one? Not necessarily high prevalence of the population, but high genetic correlation with the disease. High genetic correlation with the disease. So the same genetic variants that influence the structure of the brain also influence the risk for the psychiatric disease. Okay, so if I increase the number of proliferative neural progenitors in the developing cortex, what happens to the cortical surface area? The radial unit hypothesis. Increase. So it increases the surface area. Okay, great. And then the last question, of course, is what are your big questions? So I hope you guys, these are only just a few big questions that obviously I focus on and am interested in, but there are many other things that you can do with this data, and I hope you guys will have some questions too. So thank you guys.